Okay, so greetings from Paris. What we're going to talk about today, and I think I need to share my screen, is let me just go back into Zoom, um, share screen, and share so that you don't just look at me. Okay, so I'm going to go back here and go like this. So we're going to talk about experiments today, and specifically factorial experiments. Um, And the idea here with a true experiment is this is when you have complete control over all aspects of the design, okay? Which is distinct from a survey because surveys are just questions that you ask of people. And the major issue with a survey is that most often you're not assigning people to groups. And that's the thing that I want to say about experiments that's just important to know. Randomization, randomly putting people into groups, is a key part of an experiment. And it doesn't need to be true randomization, it could be pseudo randomization. But the entire idea behind what an experiment is that people are randomly assigned to different conditions being part of the experiment. Okay, so in a survey, that usually isn't the case. Um, a survey, you just ask the same questions of a whole group of people. Um, Correlational research, you can't assign people into different groups because their group membership is defined by qualities that they had before they came into the experiment. For instance, whether or not they're male or female, okay, whether or not they're a Rutgers student or a Rowan student. So, true experiments, people come into the laboratory, and they have an equal probability of being in one of some number of conditions. Okay, so that's what makes a true experiment an experiment. So some fancy terms that we use in experimental psych, one of them is factors. Factors is a word that is for your independent variable. Okay, and that can be an experimental variable, such as what condition you were assigned to, or it could be a correlational variable, like your gender. So when we say that we're running a multifactorial experiment, what that means is that we have many factors that we're looking at, and that most often is the case. Whenever you are reading a paper and you see the term ANOVA come up, ANOVA is a procedure that we use to look at multiple variables at the same time. So you could have an experiment where you're looking at gender, which, is one factor, the year in college that a student is, a second factor, and the experimental condition, whether or not, for instance, they had decaf or regular coffee. Those are factors. And another term is levels. And levels refers to the particular values for an independent variable. So for instance, gender is two levels, male and female, or you could have more levels depending on whether or not you're considering transgender or non-binary. College could have four or more levels. You could have first year, second year, third year, fourth year, and more than 4 years. Um, an experimental condition could have as many levels as you have. Most often, in many experiments, it's two levels. For instance, a placebo and a drug. But you could have placebo and the drug at 10%, the drug at 20%, the drug at 30%. So really, you can have as many conditions or levels of the experimental condition as you want. So a condition is a term that refers to a group or treatment in an experiment. So those who receive a drug in a drug trial are in the experimental condition. Those that receive placebo are in the control condition. And a treatment is the condition of the experiment that entails a specific manipulation. So in most drug trials, for instance, the receiving the drug is the treatment. Okay. And that's, as opposed to just the control condition. So imagine you want to study the effects of allowing students to have one sheet of notes for use while taking exams. Okay. And you want to know whether or not this is an effective way to help students perform well on the test. Um, One solution would be what's called a one group post-test only design, where you allow all students to use notes and then assess their performance on the test. Now, you can probably see the threat to validity here, which is obvious, is that, because you're allowing all students to use the notes and then you assess their performance, how do you know that the sheet helped at all? Maybe the sheet, actually distracted them and you don't have any control condition. You don't have anything to compare it to. Okay, so this is why we need to do experiments that have control conditions. What about a post-test only design with non-equivalent control groups? Well, what you could do is compare performance of students in my method in theory class with those with another professor, like Professor Markey's course, and see whether or not, they differ. And if I used notes and Dr. Markey does not, then, and my students perform better, then that must be because the notes were better. Okay. But you can see there too that there's threats to validity. And maybe, if my students perform better, maybe it's because, um, I taught the material differently. If Professor Markey's, scores were better, maybe she teaches her class better. So without a comparison group, it's hard to say anything. Now, one thing that you could do is to give what's called a one group pre-test post-test design, where you give students the test without the cheat sheet and then allow them to take the test again with the cheat sheet. Or you might allow them to take a second exam. Let's imagine that this is a class where there's, you know, um, multiple exams. Well, the problem here is that, students get better from the first exam that they take to the second exam. Okay. So a student, just like most students who retake the SAT do better the second time, it may be the case that students who are, taking this second exam with the cheat sheet, it's not the cheat sheet that's helping. It's that they're just doing better because they know what to expect in your class by the way that, the information is presented. So there are enormous threats to validity there. So which of these designs is best? None of them. Okay. That should obviously be the point of why I told you about these different. So we must come up with better designs. Okay, but what would they look like? Better designs do one of two things. Okay, they use true within subject designs, where what we mean by within subject is that a subject is tested multiple times. Okay, so everyone experiences both the control and the treatment, and you control for order and sequence effects. Okay, because in a class where you have an exam one and an exam two, there's going to be an order effect where you do better on the second exam than on the first exam. So a better design would be to use a true within subject design or use true between subject designs where subjects experience either the control or the treatment, but who receives which is determined by randomization. Okay. Now, in the example above where you use true within subject designs, randomization is going to play a role in that too, because I'm going to randomly decide on exam one whether or not you have the cheat sheet, or on exam two you have the cheat sheet. Okay, but these two designs are a good idea, and you can also mix the two of these designs. So that provides for an even better solution. So recall that in a within participant design, participants serve as their own controls by being tested at least twice, one with a treatment and one without. But the question is, might the order of conditions influence performance? Okay. If I give you a test one time where I give you coffee with caffeine, and then the second time coffee without caffeine, so I give you decaf, you're conflating having coffee or not with the order by which you're taking the tests. This is always going to be the problem with within participant designs because of the fact that we live in a temporal world where time moves in one direction. You can't go back in time and retest people as if they've never been in the experiment before. And so one condition might pollute another. There are some order effects that simply can't be controlled for. I think I've used this in another lecture that, the Milgram experiment, you can't redo or test people twice in the Milgram experiment because after the first time that they're in it, they kind of know what this is about. So they wouldn't be influenced the way they were the second time. Okay, so you need to just be, cognizant of that being a problem um, with order effects. And then sequence effects, that the order of conditions influences the results that are obtained. So, whereas an order effect makes it so that one condition pollutes another, um, the sequence effects really refer to the idea that the order of conditions influence the results obtained. So let's imagine that we're having you take, a like I had to take a stress test to uh, to assess my heart ability, to pump blood. Now, let's imagine that they every day over the course of 7 days, they had me, go on the treadmill and run, and then they measure, your heart rate and things like that. Let's imagine that by the end of those 7 days, I might just be much better at being able to, do that exercise because I had the experience of doing it. And, so the order of conditions influences the results obtained. Order and sequence effects are really kind of the same thing. Just sometimes there's a little nuance that is worth thinking about, but it's not really important for most situations. They kind of have the same impact. Now, the big problem, of course, within participant designs have to go back to the idea that might the order or sequence of conditions influence performance. Okay. And this is what I just went over. So I don't need to go over this in much detail. So order effects, knowing what will go on in condition one influences performance in condition two. And sequence effects, an example is people getting tired over the course of time. If you test people, like on the SAT, often they give you the same sort of section multiple times. You get tired of answering those questions if you have to do them multiple times. Knowledge of the previous condition. Um, and sometimes such effects are desirable. Sometimes you want people to get tired because you want to see the mistakes that they make. But in many other situations where you want to assess how good people are at something, you don't want them getting tired in that way. So one way of solving this is to do this. So you divide your class into two groups, and in test one, group one takes the exam with the notes, whereas group two takes the exam without the notes. And then when it comes time for test two, group one that took it, you know, with the notes will take it without the notes, and then group two that took it without the notes will take it with the notes. In that way, because you have two separate groups, with the order in opposite directions, you can see whether or not there is an influence of timing on the test. And you can, in fact, control for that and subtract the effect out. And we call this counterbalancing. Okay. So you flip a coin, you create these two groups, and you end up having these two groups that you're comparing. Now, with counterbalancing, it's only good when you have a small number of conditions. It's easy when you have two conditions because then there are only two different orders. But when you have three, there are three factorial orders, so there would be six different order of conditions. A, B, C, B, C, A, C, B, C, A, B, C, B, A, and so on. Okay. And when you get four, you get 24 separate orders that you would have to test, and so on. With five, there's 120. With six, there's 720. And so it becomes ridiculous when you get up to seven. You can't, what experiment are you going to run where you have over, you know, 5,000 people in all different permutations of the different orders? It doesn't make much sense. Okay. Now, another solution would be something called block randomization, where the order of conditions is randomized, but no one condition is repeated. Okay, so imagine that you have conditions A, B, C, and D, and you run it one time at conditions in B, D, A, C. The next you run A, B, D, C. The next time you run C, B, D, A. You never run the same order twice Okay, you always choose a random order. Um, Now, I mean, that's something that people do. It's called block randomization. And but it might not be possible in all situations. So it's an okay solution, but it's not ideal. You'd rather have all the permutations, but at least it's something. Another is something called reverse counterbalancing, where you run conditions first in one order and then in the reverse order. So half the subjects see, A, B, C, D, and then half the other subjects see D, C, B, A, and you just do this. You don't worry about the other permutations. Um, I mean, that's a solution too. It's not ideal, but again, the recommendation I have for all of you, if you ever do experimental work, is to have as few conditions as possible, because the more conditions you have, the more complicated these things become. And then there's sort of more complicated ways, something called a Latin square, but that requires a large number of participants, and it's beyond the what you need to know in this course. So my advice is always to try to design experiments that have as few conditions as possible so that you avoid most of these problems. Because after two conditions, you're going to need to have a much more sophisticated understanding of statistics as well. So, beyond just an ANOVA, when you have more than two conditions, you need to know about Tukey HSD, planned contrast, Bonferroni adjustments of alpha, and this is definitely sort of like a, more advanced understanding of statistics that you would need. Um, but, when you do have more than two conditions, take care to randomize, just take that advice. What about between participant designs? Um, uh, Remember that within participant designs, participants serve as their own control. But so many experiments would have such significant sequence effects that you can't have people serve as their own controls. Um, for instance, I mentioned Milgram's, but the medical experiments, you can't run people in some experiments twice because people either get better or they die. In a cancer trial, it could be the case that you don't have the opportunity to run people multiple times, or in very long, boring experiments like some of my own, where even convincing people to do some of these experiments twice would be difficult. Okay, so randomization is a key to between participant designs. You need to randomize people into condition. Okay. Um, and the major limitation is that even if you randomize people in the different conditions, did just randomness result in groups that are different in some ways that you didn't expect or, see ahead of time? And so these are sort of major and limitations of these designs. So, you can have a between participant design where you have two conditions or more than two conditions. You could have, you know, uh, control condition of decaf coffee, uh, condition two being 1 cup of caffeinated coffee, and condition three being 2 cups of caffeinated coffee. Okay. So these are all possibilities that, are um, possible to do. Now, again, as within participant design, it's still true that the smallest number of conditions is desirable because when you increase the number of conditions, you have to increase the number of participants that are required. And that's often difficult or expensive. I mean, this past spring, I had to run an experiment that, had two conditions and we needed 100 people. Getting 100 people to come to my laboratory and participate in an experiment is often difficult. It is highly annoying. It's expensive. You have to pay people to come into the lab. And so it ends up being a big challenge. So I warn people about, you know, the nature of doing this, um, that it can be difficult. And if you need lots of people, that can get expensive and time consuming. I mean, you need to hire RAs to run these experiments, or you have to run them yourself, and time is money, so it becomes very difficult. Um, and the other issue is that the more groups you have, the more potential there is for just differences in the group composition to influence results, which is the one thing that you don't want. So keep it in mind is I always say to people who are getting into experimental psychology, keep your experiments simple as possible. Okay, so that then you won't have to worry about all of these, issues that I'm talking about now. So some conclusions. Between participant designs have significant advantages. Subjects serve as their own control, limiting potential sampling differences in the condition, which is good. But between participant designs, there's no possibility of order or sequence effects, which is good. But there are disadvantages. Within participant design, there's always the possibility for order and sequence effects. And within between participant designs, there's always possible differences between the groups and the composition of the sample, and that can be bad, you know. And so the solution often is sometimes to combine the two, which is called a mixed multiple factor design. Um, but often there is no solution. You just need to take as much care as possible in achieving control in your experiments. So, factorial experiments are the most common in psychology, and they require the combination of at least two or more independent variables. Okay. So I did an experiment once where I walked up to dog walkers and people that were not walking their dogs, and I asked them on a scale of 1 to 10 how much they thought that, testing, um, you know, products on animals is morally reprehensible. Okay, I actually did this experiment. Of course, the point of this is to show how bad an experiment it is. Because just because someone's not walking a dog in Rittenhouse Square in Philadelphia doesn't mean they don't own a dog or they don't own a pet. They might just be going to work. Okay. And the people that are walking dogs, they might not even own a dog. They could be professional dog walkers that they walk dogs to make money. I knew a couple people that, made a pretty good living in Rittenhouse Square, which is kind of a wealthy area of Philadelphia, walking people's dogs. Okay. But here is one factor. Okay. And that's a between participant factor. Okay. Because you either are a dog walker or you're not a dog walker. But here, what I did is to divide dog walkers and non-dog walkers into males and females. So here, instead of just one factor, we have two factors. And what we can do is test whether or not the effect of being a dog walker or not is related to your gender. It's possible, and we can look at is what are called interaction effects. It's possible that male dog walkers, or let's call them just dog owners for now, male dog owners, really care a lot about animal testing, whereas males that don't own dogs don't care at all about the moral reprehensibility. Whereas females, maybe all females, whether they're walking dogs or not, think that animal testing is morally reprehensible. Okay, so we can test whether or not there are what we call main effects, which is ignoring male or female, whether dog walker or not dog walkers differ, or, you know, we could also just look at the main, what we call a main effect of being male or female, whether that influences people's opinions of being animal testing being morally reprehensible. We can, what we call this is collapse across variables in order to see what might be, going on here. Now, we could, of course, do this differently. We can go around and ask males and females who are dog walkers or dog, let's call them dog owners for now, dog owners or not dog owners or cat owners, maybe people who own cats are different in some way than people that own dogs. And here we would call this a 2 by 3 factorial design. Okay, because you have two, male versus female, versus three, dog walkers, non-dog walkers, and cat owners, and you're comparing that effect. Now, we can extend this into 3 dimensions. Okay, we could add an independent variable. in which case we would have males and females, dog walkers, not dog walkers, and we could see if people are single or married, whether or not marital status makes a difference. Okay. And in this case, we have sort of instead of squares that we put data in, we have these boxes or bins that we put data in. And that's how we sort of get, you know, more factors. Now, I can't draw it on a computer screen because we live in a three-dimensional world, so I can't have four dimensions, but you can have four factorial. Math works in as many dimensions as you need it to. So we're going to have a four-factor experiment where you're looking at males, females, dog walker, non-dog walker, married, unmarried, divorced, or if it you did the experiment in the morning or in the evening, okay, and asked people how morally reprehensible on a scale of 1 to 10 is using animals for medical research. Now you could even go crazier and do a 2 by 2 by 3 by 2 by 2 by 4 by 4 by 3 by 2 by 3 factorial design, where you look at male, female, dog walker, not dog walker, married, unmarried, divorced, morning, evening, smoker, non-smoker, Caucasian, Black, Asian, Hispanic, income level, vegetarian, vegan, meat eater, homosexual, heterosexual, blue eyes, brown eyes, hazel eyes, and look at the effect on the dependent variable of how morally reprehensible is using animals for medical research. Now, of course, I only give this as an example because you don't want to do this. And why don't you want to do this? Apart from using archaic terms like homosexual and heterosexual, um, you know, I, you know, this particular slide comes from a different part of my career. You don't want to do this because, in order to do this right, you would need so much data. You would need so many participants to be in this experiment because, you know, the number of conditions, it's all mathematics. So you multiply. In a two by two design, there's four conditions. And in a two by three design, there's six conditions. In a two by two design, you have eight possible conditions. In a 10 by 10 by 10 design, you have 1,000. Okay. So, you need data for each. And in our situation, we're dealing with people. We need to go up to lots of people in Rittenhouse Square in order to get those kinds of numbers. Okay. So, for instance, in that 2 by 2 by 3 by 2 by 2 by 4 by 4 by 3 by 2 by 3, you have 13,824 conditions. You would need to have data for each of those conditions, So you would need at least 13,824 people in your study just to have one person in each condition. But how easy do you think it's going to be to find a female dog walker in the morning who is divorced, non-smoker, Asian, with an income greater than 100,000, who's also vegan and gay and has hazel eyes? You know, I mean, so you would need to collect an enormous amount of people. So this is why we don't do things like this. Okay. We again, keep your study as simple as possible because the simpler your design is, the fewer data points that you need in order to make any kinds of claims. So the answer is the probability is low. You'd have to wait around a bit. So there's also tons of interaction effects. Okay, and we'll get into that in a second. In that kind of design, there's 1,024 possible interactions. Um, and interpreting interactions in what essentially is 10-dimensional space is basically impossible. So stick to two, three, or at most four factors in your research. Okay. So in factorial experiments, main effects refer to the effect of one independent variable averaged over all levels of another. I've used that term main effect already. Um, and let me just show you what this means. The main effect for gender is whether or not, when you ask people on a scale of 1 to 10, whether or not males and females differ, regardless of whether or not they are dog walkers. Okay? And that would be the main effect for gender. The main effect for dog walking is ignoring whether or not you're male or female, whether or not dog walkers or non-dog walkers differ in their answer to that question, regardless of whether they're male or female. Okay. And so in the data that I collected, this is the results. Males scored a 7, females about a 9. And dog walkers seem to think that animal testing was less morally reprehensible than non-dog walkers. But then you have to look at the interaction effect because most of the time, interaction effects are what we're looking for in science. We're very rarely looking for main effects in research. We're looking for whether or not one independent variable depends upon um, the dependent variable depends upon another level of an independent variable in influencing the dependent variable. Let me say that again. If you just read this sentence, interactions occur when one independent variable depends upon the level of another independent variable in influencing the dependent variable. Okay. So interactions are best visualized. This is why you don't want to have four-dimensional interactions. It looks a little bit like this. Is there a difference between male dog walkers and male non-dog walkers, female dog walkers, female not dog walkers? Okay. Is there a pattern within that could be significant? And look at this. Okay. For males, being a dog walker or not a dog walker really influences the degree to which you think that animal testing is morally reprehensible. For males, you know, the being a dog walker is makes it so that people think that animal testing is less morally reprehensible than people that were not dog walkers. You'd expect it to be the opposite, right? That dog walkers would love dogs, but then again, there's so many problems with this study that you can't glean anything from these data. But look at females. There's not that much difference between female dog walkers and not dog walkers on their response to the question, do you think that animal testing is morally reprehensible? Okay. So you could say here there's an interaction effect that for males, being a dog walker really affects, how they believe about the moral reprehensibility of animal testing, whereas for females, it doesn't matter so much. Okay. And this is another way of visualizing it. This is why they call it an interaction because they interact with each other. So you can see how the two lines like interact, they cross over. Um, and in reality, um, interactions are really the heart of experiments. And most experiments really aim to discover interactions as opposed to main effects. Um, you know, and they help eliminate the possibility of confounding factors. Okay, so I did an experiment with infants, and my purpose was to determine whether infants encode absolute length versus relative length. Um, and I did this by looking at their looking time in an experiment in which I showed them sticks that vary in size. Doesn't really matter for the purposes of this class. I'm going to use a different example, but in this experiment, um, what we did is we got babies bored at looking at the one over here that we called familiar. Then we showed them one that was called novel, and it's a different in these two conditions. They're different in nature. In the same ratio condition, the novel Um, and these are basically, just think of these as being liquid in glass. Okay. They're actually a wooden stick in glass, but it looks like a level of liquid in a clear glass. Okay. So that white part's just the glass part. You see how in the familiar and novel condition in the same ratio condition, um, the stick changes size. It doubles in size, right? But I also increase the size of the container. Okay, so you know, back behind the puppet stage that I show these stimuli on, I have, both of these. And then in the other condition, look what happens. I change the size of the glass, but not the amount in it. Okay, so just look perceptually at that image there. Which of those two do you think look more the same, just to you? Um, you know, if you encode information proportionally, I would say that, it's the top one, you would say that those look the same. But, if you encode absolute amount, you might think that the one on the bottom is the same. But the proportion changes in that one, whereas the proportion remains the same in the top one. Point is this, is that in the same ratio condition, infants look just as long at the familiar versus the novel one. Okay. Whereas in the same extent condition, the second condition, when we switched the, the amount to a novel amount, they suddenly are looking much longer at that novel amount because it looks to them as something new. So whereas in the top condition, It babies don't notice those two things as being different. However, in the bottom condition, they look at those two things as being very different. Okay. Um, just as an example to show you how in an experiment how important interactions are. Okay. Now, there's two kinds of interactions. Synergistic interactions are those in which the two independent variables reinforce each other's effects. And then antagonistic interactions are those in which the two independent variables reverse each other's effects. So this is an example of a synergistic reaction and that should say level at the bottom there, but it says Levick. But whereas in level one, you know, of the independent variable, the two are the same. But then in level two, being in level two of the independent variable at the bottom really, boom, increases, um, the effect of level two in the second independent variable. And that's called synergistic interaction. Kind of looks like this when you look at it like lines. And then there's antagonistic interactions where they reverse the effect. So you can see how the effect in Levick 1 is the opposite of the effect in level 2, and that they reverse each other. And they sometimes call this a crossover interaction because they cross over. And that's kind of like the strongest interaction effect. That doesn't always happen, but that's what is the strongest one to see. So factorial experiments can have all within participant factors, all between, or a mixture of the two. Okay. Mixed designs are by far most common, where you might have something like gender and, the whether or not someone is randomly assigned to, a condition, caffeine or no caffeine. Um, and the reason is that mixed designs have all the advantages of both within and between participant designs. And we talked about what some of those were. But the problem is that mixed designs also have all the disadvantages of both within and between participant designs. So what do? You just sigh and you try to do the best research that you can possibly do. Anyway, that's what experiments are about. I'll assign, maybe a basic experiment reading. And yeah, so uh, uh, what I think that we're going to be talking about next is going to be the limitations of science. So we'll get there in a minute.